Hello, and thank you for joining the fourth of our four-part webinar series entitled Bioprocess Workflows That Win. We're excited to share with you today what differentiates Repligen as a solutions provider in the bioprocessing market and the benefits of the impactful technology solutions that we bring to this market. Before we delve into the topic on innovative and differentiated technologies for the bioprocessing of new modalities today, I want to make a short introduction to Repligen capabilities as a whole. Repligen has a track record of developing, launching, and supporting disruptive technologies. We've brought 10 novel technology solutions to the market since 2017, leading to an impressive 5-year growth rate of 41%. In general, our technology platforms bring efficiency and productivity gains to biomanufacturing. We've developed technology leadership in multiple areas, including upstream intensification with Accel ATF and CrossFlow TFDF technology, downstream processing with Opus prepack columns, affinity ligands and resins, Tingenix flat sheet cassettes, and hollow fiber modules, with differentiated chromatography and tangential flow systems, with at-line and in-line advanced analytics with both our Solo VPE and Flow VPX slope spectroscopy technologies, and finally with total fluid management solutions that enable single-use bioprocessing. Technology journey has evolved to offering fully integrated solutions. We start with differentiated consumables, some of which I have mentioned, and then add standard and custom flow paths that then interface with our differentiated and scalable chromatography and TFF systems that also include advanced analytical technology for process monitoring and control. This combination of solutions renders a complete integrated technology offering for both upstream and downstream biomanufacturing. In addition, our technologies serve many markets, from traditional modalities including recombinant proteins and vaccines to new modalities including cell and gene therapy and cultivated foods. So with that introduction, some quick housekeeping tips before we move on to Shelley Parra's webinar. This webinar is being recorded for those who couldn't join us today. The on-demand video from this webinar will be available on our website at repligen.com in a few short days. Please ask any questions that you may have throughout the presentation. We always value input from our customers. We have a poll that is active now, and here would also appreciate your input. You can ask your questions and answer the poll using the Q&A and poll tabs located under the video player. With that, I'm excited to hand over to Shelley Parra, our Senior Director of Technology Positioning, who will walk us through how Repligen is differentiated in the bioprocessing of new modalities.
Hello, and thank you, Christine. Today I am excited to be able to share innovative solutions that are advancing new modality processes. So let's get started. Today for the agenda, we will start with new modality bioprocessing landscape and the challenges. I will provide Repligen solutions that significantly impact these processes, and then I'll summarize with the benefits these solutions provide in an overall workflow. Right? So if we look at the emerging modalities, they are complex and they differ based on size, type, chemistry, and their characterization is not fully known. They have unique stability issues that need to be considered, and the analytics may not be fully in place since these technologies are so new. The philosophy of one size fits all doesn't work for the production and the control of these new modalities. The industry has a promising future with these many new effective treatments in the pipeline, but that doesn't come without challenges. So if we look at the regenerative medicine and advanced therapies and how they're growing in demand and the transition from rare diseases to large patient population indications like cancer, they're increasing manufacturing scale up to 2,000 liters. So these new modalities in gene therapy alone have a very strong pipeline. There are currently over 2,000 candidates in various clinical trial phases, as noted in the graph here. And next to that is the regulatory agencies have increased approval rates of gene therapy products. So the industry is simply unable to produce enough to satisfy growing demand. Depending on what is critical for your process, there are solutions to these emerging modality challenges. Repligen can help give you a broad range of technologies and expert support to give you what you need to meet your goals, whether that's boosting productivity, optimizing recovery, reducing the cost of goods, or improving efficiency and simplifying the workflow overall. We want to help you make more product upstream and keep it in the downstream. And how are we going to do that? We will start with upstream intensification and move through the workflow. Repligen is the leader in upstream process intensification. In fact, we have over 15 years of experience in this industry. We provide two technologies for intensification: CrossFlow TFDF technology and XCell ATF technology. The first one is the well-adopted XCell ATF, which is used for diverse modalities, including recombinant proteins, monoclonal antibodies, and plasmid DNA. The second, which is the one I will focus on today, is the newer CrossFlow TFDF technology, which can be used for larger molecules such as viral vectors, exosomes, and cells on microcarriers. My colleague, Melissa Carpio, focused on these upstream intensification technologies in our first webinar of the series. For today, I'm going to focus on TFDF technology on a very high level. Right, so we'll focus on CrossFlow TFDF technology, which enables upstream process intensification of viral vectors. We can use this technique for upstream unit operations like N-1 or end-stage production intensification and continuous harvest. To understand the benefit of TFDF technology, it's good to understand the technology itself. So here shown is an expanded view of the filter, and it really shows the technology of bringing together the benefits of tangential flow and depth filtration to really provide an impactful solution. The 2 to 5 micron pore size of the filter enables cells to be retained and continuously recirculated back into the bioreactor, while the viral vector or exosomes can pass through the filter and be continuously harvested on the permeate side. An example here, this is a case study with CrossFlow TFDF, which radically improves viral vector productivity. In this case, we worked with iBet and McGill. So iBet, we worked on AAV. This was a transient transfection and lysis process, and we were able to see 3x increase in AAV production. On the other side, lentivirus with McGill, they had a stable cell line, and we were able to do multiple harvests because this is lentivirus is an extracellular molecule. So with McGill, we were able to see 25 to 30 times improvements in yield for the lentivirus process. So really, really intensified upstream from the cell growth to the harvest and through clarification to simplify that process. All right, so today we're talking about efficiency gains in the total workflow. However, there are differences between the upstream and the downstream. As we've just seen, upstream naturally provides the most potential to gain orders of magnitude more product. But downstream unit operations are diverse and require different fit-for-purpose approach and connected solutions. And we'll start with chromatography and how our integrated solutions can provide significant efficiency gains. Right? So I'll start with our Abbott Pure Affinity resins. As you know, Repligen is the largest manufacturer of Protein A ligands in the industry. As part of the Avitide acquisition, we have now expanded into affinity resins with diverse ligands, provide solutions for challenging molecules like viral vectors, exosomes, mRNA, and allow for platform purification like the monoclonal antibody industry. Our affinity technology allows for purpose-built ligands that are available commercially as catalog items, like the AAV ones we're going to talk about in a second, and then wider applications resins for customed and challenging or unique epitopes. One of the biggest differences that makes our technology unique is the 50 plus diverse scaffolds that we use to screen for bioprocess needs, whether that's high dynamic binding capacity, sodium hydroxide stability, reusability, and as well as high recombinant expression to make sure that we can manufacture these cost effectively as well. So ligands can be based on peptides or small recombinant proteins, and the attributes are suitable for bioprocessing, and that's what we design in. So we're looking for that best performance, best manufacturability. So how can affinity resins, for example, help AAV manufacturing meet clinical demand? So we offer catalog resins for AAV2, 8, and 9. Today I'm going to give an example for AAV9. In this example, we wanted to evaluate the reuse across 30 cycles with a 30-minute CIP with 0.5 molar hydroxide after each cycle. We loaded AAV feed every 5th cycle, followed by 4 mock runs of buffer load. And what we found was that we had very good, consistent and robust performance across the entire 30 cycles with hydroxide. Recovery was well over 95%. HTP and DNA removal were both similar across both, and we saw an average of 5 log for HTP removal and 3.4 log for DNA removal. So very consistent performance across that entire usable, reuse of 30 cycles. Next, I want to talk about our Opus pre-packed columns. So we offer the widest range of diameters and bed heights in the industry. This technology is platformable from process development to GMP manufacturing, from 50 microliters to 150 liters. Due to the speed to clinic that's needed for new modalities, having a single configurable and reproducible platform that can be adopted quickly is highly beneficial. We have one complete platform to fit the industry's new and growing modalities in the market, and we address critical considerations, safety, cost, and time to market, along with the meeting the demand, flexibility, and consistency that's needed. On the chromatography side, the use of small volume, high throughput screening tools will help you rapidly identify the best process conditions among a huge number of combinations between resin, type, pH, additives, a number of chromatography conditions, and then allow you to take that optimized process and scale it through the Opus technologies. I'm going to give an example. Here's an example of high throughput screening with Opus RoboColumns. These were 200 microliter RoboColumns used with a TCAN robotic liquid handler. In this design of experiment study, we looked at the separation between empty and full capsids on AAV to optimize the process. So we looked at 7 anion exchange resins, 5 wash conditions and elution conditions varying the pH and the salt types, and a total of 70 chromatography runs done in less than 10 hours with high throughput screening. So now you can develop your process in days, not months. That's a huge advantage to come up with an optimized process quickly. Right. And before I move on, I'd like to speak a bit more about the OPUS large scale for GMP manufacturing. The main drivers for gene therapy companies and new modalities is to move toward pre-packed columns are one, speed of implementation and the manufacturing debottlenecking, the lack of packing expertise in manufacturing, and quality and GMP ready solutions. Opus is a resin agnostic platform, and we bring greater than 10 years of experience packing columns reproducibly for all new modality processes. So it can be very beneficial to speed up the process for new modalities into manufacturing. As Christine noted in the introduction, Repligen provides integrated solutions, and to round out chromatography, we have use state-of-the-art innovation and precision engineering to bring a family of chromatography systems to the market from a 1/4 inch to 1 inch systems. These have been designed to maximize process efficiency for the needs of new modalities and traditional MABs. And with challenges like low recovery, growing manufacturing cost, and faster go-to-market timelines, these new modalities need modern technology designed to address those needs. Our goal is to provide effortless scale-up, maximize recovery, and offer single-use and simplified flow paths to reduce risk in manufacturing. I do not have time to expand on all the attributes and benefits of the systems, but I would like to explain a bit on how product recovery is improved and bioactivity is maintained with these systems, since that is important with emerging modalities. So here you can see how is that improved, and it's the process control, the optimal flow path, and the hardware design that allows for gradient accuracy less than 2%. And then it's the combination of injection molded tubing, over molded connections, and valve blocks that allow for removal of dead legs, allow gentle flow conditioning, and minimizing that holdup volume. Now, as an example, here's a case study from Forge Biologics, who scaled AAV chromatography from bench to a Chrome 10 chromatography system in production. They tested the scalability and the reproducibility from those scales. And what we saw was that they were able to successfully repeat the scale up, and they got better recovery in this case due to the improved performance, as I discussed on the previous slide. All right, to summarize the chromatography, Repligen offers multiple technologies that can create efficiency in the chromatography unit operation. In order to achieve step change improvement required in today's purification applications, Repligen has developed or acquired multiple new technologies, from affinity resins to platform of pre-packed columns and a family of state-of-the-art chromatography systems. Next, I'll move on to tangential flow and the solutions that we provide in this technology. All right, acquisitions made Repligen unique in that it was the only provider in the industry that could offer both hollow fiber and flat sheet technologies. So 2 different formats of TFF with unique fluid dynamics. And hollow fiber, for example, provides laminar flow with more open channel design, allowing for a gentler processing for shear sensitive molecules, whereas flat sheets allow for high mass transfer and turbulent flow and higher flux rates. Shown in this diagram is how different biomolecules typically we would use flat sheet versus hollow fiber. As you can see, whole cells, lentivirus, envelope viruses are more towards the hollow fiber, where they're more shear sensitive, compared to recombinant proteins, MABs, ADCs, AAV, towards the flat sheets, where they can take advantage of that high flux rates and go faster. So benefits to being able to have both flat sheet or hollow fiber technologies. In this case study, we performed, was on lentivirus, And in many cases, TFF steps are not optimized prior to transferring into manufacturing. But there is a clear benefit when these unit operations are optimized. So we have trained field application scientists that will guide you through the entire optimization process and scale up into manufacturing to make sure that you have a well-designed TFF step. We typically follow 3 steps. First is the selection of the TFF format. In this case, as I noted, lentivirus typically does better with hollow fiber due to the sheer sensitivity. And so viral inactivity was much improved with the hollow fibers in this case. Next, the process, it was really important to remove certain HCPs. So we looked at different pore sizes and chose the 750 KD based on better impurity removal. All of them had very good lentivirus recovery. So we moved forward with the 750 KD. And then last is around optimizing the conditions, the flux, to make sure that we have the best process that will be scaled into manufacturing. And then we support the customers during that entire process. Right. Next, our expertise and experience with our single-use TFF systems are extensive. From the bench to process scale, we provide scalable systems from product development to manufacturing. That are compatible with our membrane technology and flat sheet and hollow fiber. We provide sterile single-use processing flow paths, referred to as ProConnex, for all of these systems to greatly enhance that scale up and the GMP manufacturing needs. We've developed a complete TFF system similar to the chromatography systems that I've already discussed. So I just wanted to expand a little bit on the benefits of the system with our newest TFF system. So the CrossFlow RS20 TFF system was designed for those who work on newer modalities. It's a single-use TFF system that enables you to recover more through lower hold-up volumes, extended automation, and superior mixing, similar to the innovative designs of the Chrome system. It was purpose-built to address today's challenges, specifically with emerging modalities where every drop counts. Okay. And to summarize the TFF solutions, Repligen provides a complete TFF integrated solution from Spectrum hollow fiber modules and TangenX flat sheet cassettes to configurable single-use, ready-to-operate flow paths in state-of-the-art systems with inline analytics and technical support that will provide that can support that implementation across the entire optimization process. So regardless of modality, molecule size, process stage, or filtration goal, Repligen has a TFF solution for you. Lastly, we will walk through our advanced process analytics solutions and how this technology can be beneficial for new modalities. All right, so traditional UV spectroscopy is based on the Beer-Lambert law, where the path length is fixed and the concentration is variable. Your path length is fixed because you're using a cuvette for your sample measurement, and your concentration is variable because you typically have to dilute a number of different dilutions to figure out that linear range of the spectrophotometer. With the solo VP, however, it works with the same principle for Beer-Lambert law. However, the solo VPE path length is a variable value and capturing multiple absorbance measurements in under 1 minute. So the concentration is a fixed value and does not require sample dilution. The solo VPE system measures at 10 different path lengths. So rather than relying on a single absorbent value, you get a much broader range of measurements. The solo VPE system requires all measurements have an R-squared greater than 0.99 to be considered valid, and ensures the accuracy of the technology. So both the at-line Solo VPE and the Flow VPX systems utilize this variable pathlink technology, and the method is the same. So where they vary is how they capture their acquisition reading and the timing. So both systems have been used in various applications, and I'm going to give an example here. In this case study, we did this with PTC Therapeutics on AAV, and we wanted to look at in-process samples that, and then compare them to standard assays used now to determine genome titer and capsid titer. So the quantitative PCR, qPCR, and ELISA for the capsid titer. So samples were pulled at various purification steps, from TFF to affinity and polish chromatography, and they tested both on the Solo VPE, the qPCR, and the ELISA, and then compared those. And you can see both assays. We demonstrated fair agreement between the analytical methods used. This work is not done to replace the methods, but rather have a very fast method for in-process determination. To further this work, in-house AAV process intermediates that we do are held as a results until they come back from QC. So there's typically a long turnaround time and a backlog for lab testing. So having a very fast, reliable method that compares can be very beneficial for in-process testing. So Solo VPE is very quick. And in this case, we wanted to look at full capsids versus empty capsids or total capsids, and then compare those. So Solo VPE really gave us the opportunity for quick, direct, total viral vector analysis during development to enhance the throughput and improve decision-making along the way. All right, so in summary, to summarize the solutions and case studies we discussed today, I'd now like to show the impact of innovative tools for new modality bioprocessing to develop a bioprocess workflow that wins, right? Our main goal. So as shown throughout this presentation, Repligen addresses new modality process challenges with innovative solutions. And in upstream, the Crossflow TFDF and XL significantly improve productivity for AAV, lentivirus, pDNA, and exosome recovery. In the downstream side, our innovative solutions in chromatography and tangential flow provide a 2x downstream yield improvement, improve purity, and enhance that reproducibility, address shear sensitivity, and provide scalable solutions. And then lastly, throughout the entire process, single-use fluid management and analytical solutions improve that product consistency and quality and provide rapid data for in-process testing. So with these solutions, you can do more. In summary, you can transform your process with high-impact integrated solutions to design a bioprocess workflow that wins. We want to be your partner in this development process, and hopefully some of these solutions have sparked an interest and you'll want to hear more about this. And I want to thank you, and I would like to take any questions that you may have.
Thank you, Shelley. That was very informative. I think you highlighted the hardware and consumable technology solutions available for both upstream and downstream intensification. So we have quite a few questions that have come in from the audience as you were speaking. So maybe we can work through those. I'm going to try and put them together maybe by context, and maybe we can start with upstream and then think about transitioning to downstream.
Sounds good.
So excellent. So the first question is the introduction of TFDF for upstream used to cycle the cells with new media consistently and cycling out spent media as a way for media refresh?
Exactly. So that's exactly what it's used for. So it's a cell retention device in this case, where it's retaining the cells, and fresh media is constantly being added, the spent media being removed, and as a way to refresh it and keep that media, the cell culture, having the additives in there that are needed to keep it viable.
Exactly. Thank you. And you had talked about some of the results with the intensification of AAV viral vector production. And has that work been published?
Yes. So that work was published back in November of 2022, and it is in Frontiers in Bioengineering and Biotechnology. And we will make sure that that link gets posted to our website as well. So yes, it has been published recently.
Excellent. And you had spoken about the opportunity for intensifying plasmid production, right? Plasmid DNA production. So could you maybe talk about recommendation for ATF in that application in contrast to crossflow TFDF?
Sure. So for intensification of plasmid DNA, obviously it's for an E. coli microbial process, which in the past has not been something where intensification was needed. It wasn't really considered. It was more cost prohibitive at that point. But because of the demand for pDNA and the need for having more manufacturing facilities to making it, having an intensified process of E. coli has seen a lot of renewed interest. So between our two technologies, ATF is the better solution, mainly due to the E. coli size. So E. coli is about 1 to 2 micron, and the TFDF pore size is 2 to 5. So not really ideal for retaining E. coli, whereas the ATF is typically a 0.2 micron, which works really well for retaining the E. coli. So that's why we recommend ATF for that particular application. Yeah.
That's excellent. Thank you. And another upstream related question. So how about general experience with intensifying mRNA processes aside from the plasmid DNA?
Sure. So the mRNA process doesn't really have an upstream part of it, like a mAb or a viral vector. So the plasmid DNA, as you noted, is the starting material, and then it goes more through a downstream process. So we for our processes on how we can intensify, that would be around our OPUS pre-packed columns, using our PD for the robocolumns to in high throughput screening mode to find the best conditions to optimize for that, and optimizing for the TFF side of things, as well as the single use and the smart systems that we talked about throughout that have the analytics in place. All of those together really help intensify the process as well. Yeah.
Excellent. Okay, so I think maybe we'll transition to the downstream topics and maybe start with the chromatography aspects.
Okay.
So thinking about our affinity resin solutions and development capability, is there an opportunity for evaluating affinity resins that might be specific to exosomes?
Good question. So our affinity resins, we are definitely for the custom piece. We're always working on different modalities that these bind to, but also for future catalog resins. And exosomes is one of those ones that we are working on. So there's definitely opportunities for collaborating, beta testing new affinity ligands that are in the pipeline. And if you are interested in that, I would definitely recommend reaching out. We have an email specifically for this, and it's resins@repligen.com. Again, resins@repligen.com. But I'll also point out that all of our products, as we try to work on new solutions, we are always looking for collaborators for new technologies and new modalities with our products. So if there is something else that you heard about today that you might be interested in collaborating on, by all means, please reach out. With the chat on here or via our website as well.
Great. And thinking about Opus pre-pack columns, right? The range from robo columns to 80 centimeter diameter columns. How many different types of chromatography resins does Repligen have experience packing?
Excellent. So for the Opus columns, definitely a very wide range. And that's one of the benefits of Opus, because we will pack all different kinds of resins. So for Opus PD, these are the small robo columns, mini chrome columns, the small scale, those are well over 300 different types of resins. Where for the large scale, the GMP columns, we have packed over 250 different types of resins and about 60 resin families. So over a wide range of different types of bead sizes, as well as chemistries for the beads. Yep.
Excellent. And would those pre-pack columns be considered a single-use technology or a device that could be reused?
Good question. So for Opus, they can definitely be used single use, and we do have customers that use them as single use devices all the time. But they can also be used as a campaign, where they're just used for a few times and then discarded. Or we have customers where maybe their product isn't manufactured frequently, so they can keep those and reuse them over a number of years as well. So a wide range. So they can be single use or kind of just a campaign use as well. And Christine, if at any point if you want to expand, please go ahead.
Yeah, I was just going to say that it's I think that's the beauty, that the pre-pack column is equivalent to a self-pack column, right? So how utilized is dependent on the application, right, at hand.
Exactly.
So quite flexible in terms of the format and the packing service that's provided.
Mm-hmm.
Okay. So maybe transitioning to another question on the affinity resins. So have there been any studies done to assess the robustness of the affinity resin for viral clearance?
For viral clearance. Honestly, I cannot say for our affinity resins specifically if those have been tested for viral clearance. That's something that we can look at. I know that in the industry, there's been some debate on whether you need to do viral clearance for the AAV processes or not, but I cannot say for sure whether we have done that on our resin. So I would have to go back and look and see that. I don't know, Christine, if you are aware.
Yeah, I believe it has not been, but is planned, right? So we can address that question specifically with the individual that asked after the webinar. Okay, so maybe thinking about the TFF side of downstream processing. So for coming back to mRNA processing, right, is there a specific TFF technology that is recommended?
Okay, so for mRNA, we actually have customers using both technologies, so both the hollow fiber and the flat sheet cassettes in manufacturing. It will, for the mRNA, it depends on a few things, as with any molecule, but we have seen both flat sheet and hollow fiber work well. So we typically have a conversation with the customer, understand maybe a little bit more about the mRNA, how shear sensitive it may be, or other stability issues that we need to take into consideration, as well as what would be the preferred use at their company, because sometimes people do have prefer one technology over the other. So we work with them on what might be best for their facility, their process, and their molecule. But we have seen good performance on both. And depending on what their goals are, one may be just more beneficial over the other. So we'll have those discussions and kind of figure that out.
Excellent. Yeah, so maybe that's maybe time for one more question, right? And again, if we have not addressed any question answer, we will reach out individually or personally and respond to the inquiry asked. So if someone's interested in one or more of the technology offerings, Shelley, right, how do they get started? How do they learn more after the information that you've provided today?
Yeah, I'm hoping, I know there wasn't a whole lot of detail in all of this because we were trying to get so much out there, but if there's anything that was interesting to you that you want to hear more, my recommendation is to reach out to your local account manager. If you don't know who your local account manager is for Repligen Technologies, you can contact us via our website and ask those questions, and we will get back to you. So then we would have come in and have a more in-depth technical conversation, understand your needs, your goals, and go from there. Support and evaluation and all of that. So, yeah.
Excellent. Thank you. So with that, I just want to provide a reminder that all of the content from the four webinars, that we've run through the month of February are available with on-demand content on our website at repligen.com. And I also want to thank you for your time today. We hope you found this particular webinar to be valuable, and we look forward to following up with you on future opportunities. Thank you.